going to continue our discussion of molecular genetics today. Today looking at how the information in the DNA actually becomes the organism itself. So if we look at the DNA double helix again remembering that it's composed of two strands of DNA and a nucleotide sequence. Again, what we're going to be interested in then is how does this nucleotide sequence of DNA essentially become an organism? So in the past, we've talked about this relationship. So the DNA nucleotide sequence then translates to an amino acid sequence of a protein, which determines protein shape, which determines protein function. So we're going to need, and this is what's known as the central dogma, but we're going to need to refine this a little bit in that really what DNA does is it doesn't directly code for proteins, it needs an intermediary. And that intermediary is going to be RNA. So what actually happens then is that DNA codes for RNA. And I've kind of pluralized that because there are RNAs that have different functions, but it all has the same basic chemical composition. And this is a process known as transcription. Transcription occurs within the nucleus, so that'll occur in the nucleoplasm. Then these RNAs then will diffuse into the cytoplasm of the cell and will work together to produce a protein. And this is a process known as translation. So the RNAs work together through the process of translation to generate proteins. And that occurs in the cytoplasm of the cell outside of the nucleus. Now the proteins then will either become part of the structure of the cell or the organism. They'll do work. In other words, they might be enzymes or transport molecules, these kinds of things. Or they may be regulatory, meaning that they will feed back potentially on RNA or DNA, affecting the function of the DNA and RNA, determining whether a gene will function or not. So that's where we're headed. So we're going to be looking at this process of transcription and translation and how proteins are produced, because it's the proteins in that do the work of making an organism or becoming the substance of the organism. All right, so we need to know a little bit about RNA. So RNA stands for ribonucleic acid. So the R in A comes from those terms. And RNA is a nucleic acid. So it's very similar to DNA, but there are a few structural differences. So again, we're looking at strands of nucleotides. So the nucleotide will be the basic unit again. And so we'll have a phosphate group, a sugar, and a nitrogenous base. So first let's look at the sugar. So one of the differences between DNA and RNA is that RNA will use a different sugar, and also there'll be a slight differences in the nitrogenous bases that are used. So again, first looking at the sugar, so RNA uses the sugar ribose, so it's ribonucleic acid instead of deoxyribonucleic acid. And just as in DNA, the ribose sugar has the carbons numbered, so we'll have our 1, 2, 3, 4, and 5 prime carbons. So that means then that when we look at an RNA strand, again, we can work from 5 to 4 to 3, 5 to 4 to 3, 5 to 4 to 3, all the way down. And so we'll have a 3' end of RNA strands and a 5' end of RNA strands. And so this, again, is very important because it is the sequence of the nucleotides that eventually determines the amino acid sequence, protein shape, and function. So we do have to keep track of 5' and 3' ends. Now, another chemical difference between DNA and RNA then also is in the nitrogenous bases. So whereas DNA uses the pyrimidines, cytosine, and thymine, RNA is going to be using the pyrimidines, cytosine, and uracil, and both DNA and RNA both use adenine and guanine. So the difference is that instead of thymine, RNA is going to be using uracil. So when we look at our base pairings in DNA, adenine always pairs with thymine, but in RNA, adenine will pair up with uracil. So that will be the complementary pair there. And then they'll both be pairing guanine and cytosine, as we've discussed before. So again, when we see uracil, we know we're dealing with a strand of RNA instead of DNA. So as we compare the differences between DNA and RNA then, DNA uses deoxyribose sugar, whereas RNA uses ribose sugar. They're both composed of nucleotides, and so they're similar in that they both use adenine, guanine, cytosine, but where they differ will be in uracil. So RNA uses uracil in the place of thymine. DNA tends to be very long. So when we look at the nucleus, say, of a human, there are 46 chromosomes in that nucleus. If you put those end to end, they would be more than 2 meters long. So they're as long as I am tall. Anyway, when you compare that to RNA, RNA is relatively short in terms of the length of their strands. So that's an important difference because that has an effect on the function. So DNA, because the chromosomes are so long, they cannot diffuse out of the nucleus. They can't get out of the pores. But RNA can. So what's going to happen here is that DNA then transcribes to RNA. RNA moves into the cytoplasm, and that's where proteins are generated, and those proteins then make the organism. And then lastly, those chemical differences also translate to some structural differences in that DNA, of course, is a double helix, 2 strands in a helical form, whereas RNA typically is single-stranded. It can be double-stranded. It's more typically single-stranded. It can fold up and form self-complements with itself, and it will take on many shapes, and it can have a variety of functions. So again, that's going to be important, and we'll talk about that in a few minutes. We'll see examples of that. So RNA then can form complements with DNA. So if we started with a DNA strand, if we were to make an RNA complement from this, again, what we would do is have to put our 5' end opposite the 3' end, 3' opposite 5'. And as the strand is being built then, opposite guanine you would have cytosines, but opposite adenines you'd have uracil. Opposite thymine adenine, guanine cytosine, cytosine guanine. But when we get to adenines, it's always going to be uracil opposite, adenine opposite thymines. So when you see uracils then, we know that we're dealing with RNA. And also I should say that even though RNA will form complements with DNA, it's not as strong as the complements that form between DNA strands. And so these RNA strands then tend to peel off and then take on a shape of their own and have some function. All right then, so again, reviewing where we're headed. We're looking at DNA, coding for different types of RNA, or at least RNAs of different nucleotide sequences, in a process of transcription taking place in the nucleus. And then those RNAs will diffuse into the cytoplasm to produce protein in a process known as translation. And those proteins will be structural, do the work of the cell, or regulate RNA and DNA. Now, there are a number of different types of RNA that are produced. In terms of protein synthesis, we're going to be interested in 3 different RNAs. One of those is known as messenger RNA. Messenger RNA has the information about the sequence of amino acids of the protein. So the messenger RNA is saying, this is the protein I want you to make. Transfer RNA, or tRNA, will be transporting amino acids. And then ribosomal RNA becomes a component of ribosomes. But we'll go ahead and look at examples of all this in just a second. So let's go ahead now and look at that first process, which is transcription. All right, so transcription then is occurring in the nucleus. And what we have then is that we're going to be making a strand of RNA. RNA is a nucleic acid. So what that means then is we need a polymerase. But instead of DNA polymerase, we need RNA polymerase. RNA polymerase will build RNA strands. So we have RNA polymerase, and RNA polymerase will bind to a chromosome at a site known as a promoter. And we'll talk more about this in future modules, but the RNA polymerase can recognize promoter regions, and then it will, quote unquote, transcribe the DNA from that point. So let's go ahead and look at what that process is all about. So RNA polymerase then will bind to the promoter region, and this is where it just kind of locks in. And then it will separate the strands of DNA and then read one of the DNA strands and make a complementary strand of RNA. So again, the polymerase will move along one of the 2 strands. And as it does so, it will match up RNA nucleotides to the existing DNA nucleotides. And as I mentioned before, that association is not particularly strong, so the RNA will eventually peel off. But do notice that where we have thymine, adenine, guanine, cytosine, and so on. But again, what the polymerase is doing is reading the existing DNA and matching up RNA complements. But where it encounters adenine, then it will be putting in uracil. Okay? So this is the process of transcription, reading one of the strands. And this is something else that's important to understand about the DNA. Only one of the strands in will have information about the protein. So this is sometimes referred to as the coding strand. But it will have that information. And the other strand has a different sequence of bases, so it's really just there as support and reinforcement of the existing strand. It's just to help to stabilize the coding strand. So at some point then, the polymerase will reach the end of the coding region and will dissociate from the chromosome. Now I want to show you another animation as well. Oh, and this is continue showing us that this will peel off. So we'll continue on, eventually dissociate, and the RNA peels off. And then that can eventually diffuse into the cytoplasm and do whatever it's supposed to do. Now, I would like to show you another animation really of the same process. And so what we're looking at here is we're inside of a cell, we're looking at the nucleus, and let's look at another view of transcription. So again, the idea is to visualize these processes as much as possible. If you can visualize them, you can describe them. If you can describe them, you can answer questions about them. So here we're looking at the chromosome, and here we have RNA polymerase, which is going to bind and begin to transcribe. And as it does so, it produces an RNA strand. So now we're getting a little closer look. So here it's reading one of the DNA strands and it's building a complement out of RNA. And we're going to consider this strand that it's making an mRNA strand. So RNA polymerase builds the strand 5 to 3, just like DNA polymerase does. So the new strand is built 5 to 3, and again, it reads existing DNA bases and matches up complementary RNA bases. All right. Okay, so that then brings us really to the next step in the process, and that involves processing. So we're looking at this strand that's being produced as being an mRNA, a messenger RNA, and the messenger RNA contains a sequence of RNA nucleotides that translate to an amino acid sequence of a protein. Now, before that second part of the process can take place, the mRNA has to be altered in some ways. And so what happens is that there are other RNAs that are known as small nuclear RNAs, which associate with other proteins to form these small nuclear ribonuclear proteins, sometimes called SNRPs. And they associate with still other proteins, and they will attach to an mRNA strand and basically then cut out sections of that RNA strand that will stay in the nucleus. So some portions of that RNA strand will stay in the nucleus, they're known as introns, and then it will splice together or other portions of the mRNA strand. Those are known as exons because those will be spliced together and those exit the nucleus and get into the cytoplasm. So we start off then with what's transcribed is what's known as a pre-mRNA. And then again, introns are spliced out of it, exons are spliced together, and this becomes the mRNA itself. And this is the strand then that has a sequence of RNA nucleotides that will correspond to a sequence of amino acids and determine the protein shape. So these are called exons because they exit the nucleus, they get into the cytoplasm, and that's where the protein is actually going to be made. The introns stay in the nucleus, basically get disassembled, and those nucleotides then can be used for further assembly of RNAs. All right, so at the end of this RNA processing, some other types of processing occur as well. But at the end of the process, then, what the RNA will have, the mRNA will have then, is what we call a GTP cap. And GTP is very much like ATP. It's an energy-containing molecule. There's going to be a sequence of nucleotides that are known as an untranslated region, so this doesn't really code for a protein. There's going to be the protein coding sequence, another untranslated region, and then what's known as a poly A tail. Lots of adenines are added at the end of the mRNA. So again, this portion here would be where the exons eventually essentially are, and then a cap of GTP, which contains energy, is put at one end, and a bunch of adenines are put at the other end in this poly A tail. So that's what the mRNA looks like at the end of processing. So let's go back to our animation then, and we'll look at an animation of this processing as well. So again, we have transcription occurring, 5 to 3 direction. The RNA strand is peeling off. The GTP cap is going to go at one end. And again, the polymerase is moving along the chromosome, producing the mRNA. And so again, here's where our spliceosomes, the small nuclear proteins with other proteins called spliceosomes, are going to bind to the mRNA strand. They're going to splice portions together. And that part that went out, if I backtrack just a little bit, then this would be the intron. This is the one that's going to stay in the nucleus. And the exons are spliced together. And then adenines are added at the end, so that's our poly A tail. And so this now is a functional mRNA, and that will diffuse into the cytoplasm where translation can occur. And that's our next step. So again, as we look at the overall process, I do want to say first, however, that when we look at the sequence of nucleotides, what's going to be significant is that the nucleotides are read in threes. So there is what's known as a triplet code. And the reason for this is that if you look at the nucleotide sequence of the mRNA and how this translates to amino acids, there are 20 amino acids but only 4 nucleotides. So if you took the 4 nucleotides one at a time, that would only give you 4 possible combinations. And that would not account for the 20 amino acids. If you took the 4 nucleotides 2 at a time, that would give you a total of 16 combinations, which still does not account for all 20 amino acids. So what happens is that the code in DNA and RNA then is 3 at a time. So we take the 4 bases 3 at a time. That gives us 64 combinations, which not only accounts for all 20 amino acids, but it gives us lots of repetition in that as well. So again, looking at the overall process, we've now talked about transcription. Now we're going to look at how different types of RNA build the protein in the process of translation. Okay, so we'll start with this animation. So what we're looking at here is the mRNA strand. And it begins with what's known as a start codon. And so what's going to happen is that a ribosome is actually going to attach to the mRNA. And this is what's known as initiation. And there's 2 subunits to a ribosome, so a small subunit and a large subunit. So the rRNAs then form part of the ribosome. And then what's coming in here is that we have what's known as a tRNA. And the tRNAs will bind to the mRNA strand. Now, I want to take just a minute and talk about the tRNAs. Because when we look at the mRNA, again, we're going to be looking at 3 bases in sequence. So this is what's known as a codon. The tRNA has an anticodon, which is complementary to that. So tRNA is nowhere to bind on the mRNA because the codon of the mRNA is complementary to the anticodon of the tRNA. And the tRNA then will carry a particular amino acid. So if we could look then, I want to take a look briefly at the structure of tRNA then. So if we go to this, so tRNA then is another type of RNA. And so again, there's going to be a 3-base sequence known as the anticodon, and it will correspond to a particular amino acid. And here's a more realistic view of what a tRNA looks like. But again, the anticodon will correspond to a particular amino acid. So there are enzymes then that will bind ATP and a particular amino acid and then attach those to a particular tRNA based on what the anticodon is. And so we end up with a tRNA that carries a specific amino acid. And so this is a process known as charging. So the tRNAs then must be charged. So going back to our animation then, and continuing on then, we have a tRNA anticodon complementary to a codon. And then the other unit, the large subunit of a ribosome, will attach. And so there are 3 sites here, an A site, a P site, and an E site. And then another tRNA will sit in the A site. So what the ribosome then is doing is kind of holding everything together. tRNAs are binding with their anticodons, complementary anticodons. That positions them for bonding. And then the mRNA will slide through the ribosome. Everything is going to shift this way. This tRNA is clipped off and can be reused, so it can be recharged. That opens up the A site for a new tRNA, and so the codon will determine what anticodon goes there, which determines what amino acid goes there. So we have a new codon complementary to an anticodon. That tRNA holds a specific amino acid. And it just keeps shifting along. So tRNAs go from the A site to the P site to the E site where they exit. And there are 3 codons out of the 64 that do not code for any kind of a tRNA or an amino acid. And so what happens is that a releasing protein, a releasing factor will sit into that spot since there's no corresponding tRNA. And then when the ribosome shifts or when the mRNA shifts, that causes release of our polypeptide unit. Okay? So the mRNA can be reused again, the ribosomal units can be reused again, and this will begin to fold up into a particular protein. Okay, now I would also like you to quickly see the Bioflix animation on this as well. And so let's go ahead and continue with this animation. So again, you might recall this is the mRNA, which has moved out into the cytoplasm and has been modified. So the small subunit of the ribosome and a tRNA will attach to the first codon. Large subunit will attach, and then subsequent tRNAs will attach carrying particular amino acids, and they will bond. And then shift, exposing a new codon, complementary to an anticodon of tRNA holding a specific amino acid, allows peptide bond to form, and so the amino acid chain begins to grow as the ribosome moves along the mRNA strand. So again, these would be complementary, and 3 successive mRNA nucleotides determines a particular amino acid. So it's showing charging of the tRNAs, and again, as the ribosome continues to work, the amino acid chain continues to grow. Okay. All right, so let's go ahead then and look at an example. If I were to give you a DNA double strand, could you translate that into an amino acid sequence? And the answer is, I hope you can. So let's see what happens. So only one of these strands is going to actually be the coding strand for DNA. So we'll say this is the coding strand. So I'm going to go ahead and eliminate the other strand. And so what would happen in transcription then is that RNA polymerase would bind to the promoter and then transcribe this strand into an RNA strand. And so the RNA polymerase would build the new strand 5 to 3, and it would transcribe an RNA strand. And again, we would have thymine adenine, but every place there's an adenine, we'd have uracil opposite that. And then that would peel off, and then a ribosome would begin to work. And so again, the ribosome is also going to work 5 to 3, and that's important because AUG is not the same word in effect as GUA in terms of this language. So that's the first codon. So let's go ahead and see what that would translate into. So the code of mRNA and DNA is universal. So this is one of the things that speaks to a common ancestry of all organisms. We all use exactly the same genetic material, and it's exactly the same code. So that AUG codes for methionine in a mouse or a bacterium or a human being or a banana. It's all the same. So if we look at our sequence then and how we use this chart then is the first codon then is A. So all of these would be codons that start with A. This column would be the second portion of the codon, so U, and they overlap here. And then for a third codon we go over here and we look at G, and that's where they all overlap. So AUG corresponds to the amino acid methionine. So that would be this first amino acid in our protein here. And as it turns out in eukaryotes, it's always the first amino acid. So it's known as the initiation codon. Then the ribosome would move along, look at the next codon. And so our next codon is G, U, A. And so that gives us the amino acid valine. And you'll notice that there's overlap in the code. So all of these code for valine, but we're interested in this particular one. So the second amino acid in our sequence is going to be valine. So then the ribosome would move to the next codon and CGG. So if we go to our chart, first codon is, or the first nucleotide is C, G, G. That translates to the amino acid arginine. So arginine would be the next in line. And also keep in mind then that the tRNA is what's actually positioning these, and so the tRNA would have an anticodon. So if this is 5' GUA, the anticodon would be 3' CAU. Okay, so the ribosome would read the next codon, and if we put it into our chart, we'll find that that translates to tyrosine. And then the next codon, UCC, U, C, and C. And so that translates to serine. And then finally, we're going to get to UGA. So again, U, G, and A. And you'll notice that that is what's known as a stop codon. So there are 3 stop codons, UGA, UAA, and UAG. Those 3 codons do not have any corresponding tRNA or amino acid. So again, what would happen there is that a so-called releasing protein, a releasing factor, would sit in the ribosome at that spot. And then when the ribosome translocated or moved to the next codon, that wouldn't have anything to attach to. So the amino acid sequence is released. This now begins to fold up into the protein and take on a specific shape. So I think you can see then that this is a pretty complex process. But what it all boils down to then is the sequence of bases. And directionality is important here. So for example, again, this worked 5 to 3. We happen to be working left to right, but that's not what's important. What's important is that we were reading the codons 5 to 3. Because if we worked in the opposite direction, we would get a completely different amino acid sequence. So it's a universal code. So again, AUG codes for methionine in a human being, a banana, a mouse, a squirrel, whatever you want to say. This is one of the things that, again, speaks very strongly to a common ancestry of organisms. And again, it allows us to work in any direction. So if you happen to know the amino acid sequence of a protein, you can work backwards to RNA sequence into DNA sequence, and then you can look at the DNA of an organism and look for specific genes. So we'll be talking a lot more about molecular genetics in the future and technology, but a lot of it boils down to the relationship of proteins and DNA. So until the next time.